Welcome to week 4 and the walkthrough of the lab. As always, click here to download the worksheet that you'll be filling in. Down over here is our rubrics and our free S-post for posting your observation tables. The link is where I presume you got the video from, and we're going to click here to get to the rubric. Let me pause and open that section. Okay, here's the week 4 rubric. Always check how much each point is worth, or each part, I guess, is worth point-wise. And you see these questions are worth a lot extra down here. So you probably want more information. You need to have that perfect title page. It's week 4, so everything should be pretty perfect in terms of title page, complete sentences, grammar, references. Spelling issues, don't have that. Just run it through a spell check. Okay, so you would, of course, to get your title page, your lab assistant will just have this, not the rubric above it. You just go to Insert, click on Page Break. That will give you a blank page. It's the easiest way to just put in that title page. Okay, plant cells in osmosis. So you're going to be soaking your potatoes, right? So you want to have your initial mass, your final mass. In the mass difference, if you went up in weight, add a plus in front of it. If it went down in weight, add a negative. Then tell me the percent of change in mass. Include that positive or negative in the change. So then the class average, if available, it is available. So make sure to have at least one other student's data in here. And then your hypothesis, you want to do as always an if-then. So if you see this, then it means this. So if is going to be what your experimental results might be, and then is what that actually means. Okay. Scroll down. We'll have a data of potato mass versus solution. This will be the change of mass versus the solution. I would probably just do the percent. Yeah, I do percent change of mass, but if you're not sure, bug me. Because I'm being lazy and not opening the answers at the moment. Okay. So define osmosis, define it, and then tell me how you saw it. So what did you see that shows osmosis? List the molar concentrations, whatever they are, where water entered the potato strips, and do this in a sentence. And then is this hypo, hyper, or isotonic? Do the same thing, but now when the water left, so this should be positive, that will be negative, right? And again, if it's hypo, hyper, or isotonic, then what was your hypothesis? Was it supported or refuted? And then let me know why. And you were asked to place the potato strips, why can I not say strips today? Potato strips in a solution that would cause the potato to neither gain or lose mass. If you were asked to do this, what molarity would you choose and why? So this is where you probably want to look at the graph and see where it goes through 0. So that might not be one of the strips you said. Let me scroll back up here. Like, let's say that happens between 6 and 8, you might say 0.7. So it doesn't have to be one of the numbers you gave. It might be in between. Look at your graph and see where it's the closest to nothing changing. Okay? So here we're talking about diffusion across the membrane. So you're going to say the color and then what you saw. And then here initial and final results, the same concept. What happened? Okay, and so now we're going to talk about what happened. So you're going to define diffusion. What is it? I'd probably give me a sentence on like if you saw it or not, but just say what it is. The IKI indicator test for the presence of what substance? Benedict's test for what substance? It will tell you that. Plus we just did that in the last week. I was wondering if it cheated and made it really easy. You have to work a little bit. Just tell me what it's testing for. So now we're looking at, in the first part of exercise 2, a glucose/starch solution was placed in dialysis tubing, and the tubing was placed in the container containing the indicator. So did your starch stay within the tubing or did it move out? You gotta tell me why and then how you saw that, right? And then the second part is gonna be the same concept, but now we're gonna heat it with our Benedicts, and then we're gonna say, does the sugar move or stay within? Okay, and you're going to give me observations, which will probably be the color change for both of these. Now, what was the purpose of testing distilled water with the Benedict's reagent? Give me the word. We learned from week 1. This is called what? And then, how is a cell membrane similar to the dialysis tubing in this experiment? What was this one called? Diffusion across a membrane. So what might it be similar to, right? I was like, a lot of them will. If you look at the title, sometimes it helps. Okay, then we went, is the movement of substances in this exercise active or passive? Did you have to put energy into them to get them to move? From your data, basically what was bigger, starch or glucose? If you're not sure, you can use an outside source to verify this, but this should be by what moved, right? And then that is the end. So of course you need a reference. I would probably use my reference. I might throw it in here. Oh, you know where I would use it? Probably the cellular membrane, and then any of the definitions, right? You have to define diffusion. That's a good place to use an outside source or the lab manual. You just need a citation. If you're paraphrasing things from the lab manual, it's good to use it. Osmosis, that's another place. Okay, so hopefully this helps make everything make sense. As always, I would read the lab manual at least once before doing the experiments, and give yourself more time than you think you need, because if there's nothing labs are better at than it is torturing people. I swear, they're there for evil. But you do learn a lot, and the hands-on, you just can't beat it, right? The hands-on. It's just sad that you can't be in a big group, which is why we share our lab data table. So if someone gets really funky data, you know, of course, sometimes the person who's the funky data is the only person with the correct data, which is why submit a draft before Monday. Okay, I hope this helped. Enjoy.